Hello, my name is Richard Lund. I'm coming to you from Rancho Varna Studio 3 here in Los Angeles. I'm on a journey to live a healthy 100 years. Why not come along with me? Today I'd like to dedicate this program to Tom Leahy. Tom was a good friend of mine. He left us from this planet and with this earth, and he's with my Father in heaven today. And he taught me a lot about things, particularly something we'll mention today. He was the one that introduced it to me. So I just wanted to say that we have to remind everyone that this is an educational program. If you're sick, you need to see a doctor, a real doctor. I am not a doctor, and I don't want you to follow my medical advice because I'm not giving any. But if you do find a doctor, I hope you find one that speaks herbal medicine. Today's presentation is called Tales from the Lab of Mice and Men. Robert Burns had a very important quote: "The best laid schemes of mice and men go off to glee." And from that, we usually have a shortened version of the best laid plans of mice and men go awry. Anyway, that's what that's about. What can we learn from mice and cell cultures? While it's true that we often study how a substance acts in a petri dish and in a mouse, the things we learn can trick us into believing that they work the same way in people. So we have to take this with a grain of salt, but it is where we start. Today, let's look at a good study that explored how the SARS-CoV-1 virus behaved, and especially what seems to be the big deal that makes the COVID-19 disease so virulent in some folks. Now, as a reminder, SARS-CoV-1 is the virus that caused the disease SARS, which appeared in 2002 and 2003 and disappeared apparently from the human population by 2004. And today we have COVID-19, which was caused by the virus called SARS-CoV-2. So let's go on from there. I read a paper from two labs, one in Madrid and one in Iowa City, Iowa. Marco L. de Diego et al. Reported on what they found out in the severe acute respiratory syndrome, that's called SARS, coronavirus-infected mice, that increases survival. It's pretty important. And perhaps what protein in the virus gives us the bad symptoms. So if we could find existing molecules that could interfere with that protein and what it does, we might keep more people out of intensive care and off the ventilators. That would flatten the treatment curve. That would be great. So here we have an illustration provided by Alyssa Eckert and Dan Higgins. Now you notice this is fairly, I suppose, well known by now. It's the SARS virus as a general illustration. You have the red pieces sticking out that are called S proteins or spike proteins. And this is what gives the SARS virus the coronavirus name as far as the shape. Then you notice on this illustration it also says M protein, which would be for membrane protein, and then E protein, which would be an envelope protein. And the E protein is illustrated in a yellow color. This is the close-up of that same illustration. You can see the E protein in yellow right in the center of this picture, just about. So these researchers made a special coronavirus-like SARS-CoV-1 that would work in a mouse. And they wanted it because they wanted it to work in a way that would be almost the same as far as the symptoms in people. Then they made another just like it, like this SARS-CoV-1 for the mouse adapted, they call it. But it was just like it, but not with the E protein. So without the protein. Sometimes it's written delta protein. And then they infected the mice. And what do they find? But first, let's talk about nuclear factor kappa-B, or NF-kappa-B, or the long name is nuclear factor kappa light chain enhancer of activated B cells. That's what it was called when it was discovered, and so today we just refer to it as NF-kappa-B. I guess my Mac doesn't have a Greek letter for kappa, so we'll just write it out. I first learned about NF-kappa-B from my friend Tom Leahy. Tom was a generous man who made some money in stocks in the 90s, actually did pretty well, and then decided to invent something to help people using natural products. His first patent was to treat a type 1 diabetic, actually a little boy about 6 years old, with a flavone called luteolin and not insulin. And he went on to work with this purified molecule that's made in plants to see if he could help people with a number of conditions like Parkinson's, Lou Gehrig's disease, and autism. And I've seen videos that he shot of people with these various conditions that were responding to the luteolin and the way he prepared it. And by a mistake in processing, he combined another flavone with the luteolin with what he said was a salt bridge, and it made it more effective by staying active in the body and delayed liver clearance. So Tom told me that this little mistake was a good thing, and the people that were trying the product after that mistake was made in the processing reported more effectiveness. Tom sat me down one day in his office and went through the biochemistry of NF-kappa-B. There are some refinements now, but the basic mechanism that he described is called the canonical pathway today. What is NF-kappa-B? It is a structure in the cytoplasm of two joined proteins that are ready to go to the nucleus when activated, but are bound up with an inhibitor. So when a receptor on the cell membrane is triggered, a molecule is released from that binding site to go to a group called, the group is attached to a molecule called NEMO, sometimes also called, IKK gamma, and it changes the binder on the NF-kappa-B dimer. A dimer just means that it's two proteins together. So it changes the binder on the NF-kappa-B dimer to release it. And that pair of proteins, then enters the nucleus and activates a number of genes, making proteins, and ultimately gets inhibited again by one of those proteins that it makes. That's the quick version. Here's a diagram from a paper that reviews inhibitors of NF-kappa-B signaling by Gilmore and M Herscovitch of Boston University. Here's the title of the paper, T.D. Gilmore and M Herscovitch. Well, that's a mouthful, isn't it? And they talk a lot about this. In fact, it's great, a very interesting title. It's Inhibitors of NF-kappa-B Signaling, 785 and counting. So there's a lot of them out there. So this is their cartoon of that. And if you take a look here, up here, there's a circle. That's more or less supposed to represent the cell membrane on the outside of the cell. Then there's an inner membrane where the nucleus is inside and the cytoplasm is out here. You notice these little kappa things travel over to NEMO, which in this case would be releasing IKK alpha, and that would travel down to the inhibitor, which is sitting on the dimer. Here it says P50 and REL-A. Now, REL-A is also called P65, and there are actually 5 of these little proteins that become part of NF-kappa-B. And what it is they have a similar structure in part of each protein and then a different structure in the other parts. But we'll go back to that some other day perhaps. So you see a couple little P's there, and that's the phosphorylation that's happening, which then allows the inhibitor to release, and the inhibitor gets disintegrated. There's ubiquitination going on. I'm not really sure what that means, but anyway, don't worry about it. We'll get there nonetheless. So the dimer travels then to the nucleus, goes through a pore in the nucleus membrane, and gets down here and then activates a gene, which releases, the messenger RNA, which then travels to the cytoplasm and goes to the ribosome and builds protein. So that's how proteins are built, basically. Activation of a gene, translation of that information, transfer to the I always get translation and I was going to say transfiguration, but that's a theological term. I'm getting off track here. We'll go back to it here. But the main thing is that this is how the cell can respond to the triggers that are out here coming up against the cell very quickly. And because there are so many different things that can trigger this NF-kappa-B signaling. Quite frankly, even viruses can trigger it, and many other things. So it's a really complex system. I mean, I'm just touching the surface of it. But it is important because we're going to be talking about that as we get into the study with the mice and the SARS-CoV-1. So, just for your information, I've got another stack over here of the all of the things that activate. This is a table from that paper. Just reams and reams of molecules that activate NF-kappa-B. So here's Apogen, and that's actually one that Tom worked with some, and it's a natural product, and it's a NF-kappa-B, probably alpha or beta upregulation. So that's what that's talking about. So I'll just take that away, and we'll look at that perhaps another day. So if we have the activation of NF-kappa-B, we're going to look at the paper itself. And I think we'll go back to talking about NF-kappa-B just a little bit first. At the time of writing the paper about NF-kappa-B, these two cataloged 785 inhibitors of NF-kappa-B, and I'm sure there are more now. And they said that some viruses, as I mentioned, activate NF-kappa-B, but even to keep it activated and to help control the timing of cell death. So one of the things that activation of NF-kappa-B can be related to is inhibiting cell death. And there are some other triggers in the cell that might push it toward deciding it's time to die. And then this activation of NF-kappa-B can inhibit that. So it's a way of controlling when the cell dies. Now, viruses need time to make all the parts of the new viruses. There aren't a lot of parts in viruses, but there are some. In this case, we have an RNA strand that needs to be made, replicated. And then we need to make a few proteins. We talked about, we had the S protein, the spike protein, we had the membrane protein, we had the envelope protein, and then there's also a nucleocapsid protein that is also made. So there are at least 4, and there may be others, but those are the ones I'm familiar with. So viruses need the time to make those parts, and then sometimes want the cell to spill out the baby viruses to go on to make more in adjacent cells. Okay, so I call it baby viruses. They're so cute. They're not cute. They're ugly. And they're not really babies. They're just progeny, or copies, really, is what it is. If you go to a copy machine, I guess some people have copy machines, don't they? You put a piece of paper on the glass, close the cover, and then you push the button for how many copies you made, and then it just cycles through. But viruses take a little bit more time than a copy machine. So anyway, by timing cell death, they can have time to make what they need. And then at some point, they might want to cause cell death, allow cell death, in order to spill out the contents, which then can go invade other cells. Crafty buggers, my Scott friend Harwood would say. So let's go back to the mice. Oh, but wait. Let's think about that E protein first. So we'll go back to our paper here. There's a lot of things that are kind of scary about SARS. We're scary about SARS because people are not infected with it now. But today, of course, we've got COVID-19. And we're in a pickle. The world is in a real world of hurt regarding this virus and the disease it causes, COVID-19. So this paper has a lot of stuff in it, and I will read through some of it, and perhaps we won't go through all of it, but I'll try to make sure that you see the points that I would like you to see. And then you may want to, get the paper because it's available without charge. Unlike some other papers I've looked at recently, I wanted to read, but they were, $40 or 40 pounds sterling or something to get them. So that's another story. So severe acute respiratory syndrome, the SARS, caused by a coronavirus that was now called the SARS-CoV-1, affected approximately 8,000 individuals during the 2002 through 2003 epidemic and had a mortality rate of 10%. Now, that's a lot of people dying, but the good thing was that it only had 8,000 people infected. Now, As of today, I'm sure we have officially more than 200,000 around the world, but of course that number is probably significantly higher. SARS-CoV has not reappeared in humans since 2004, this paper reports. Now, this paper was published January 2014, so 10 years after that. But SARS-like coronaviruses are present in bats, circulating all over the world, making another SARS outbreak a possibility. What? 6 years ago, we had a warning from the people that wrote this paper from Spain and from Iowa City, Iowa, and we didn't heed it. There's another guy, Michael Osterholm, I believe his name is, from Minnesota, who also wrote a book, came out maybe about the same time, and also said, we've got a problem, folks. We need to get ready because this is going to come and bother us. And unfortunately, these scientists that have predicted this are right. It says the fact that zoonotic coronaviruses frequently cross species barriers to infect humans makes the generation of vaccines and the identification of antivirals important goals. Yeah, now we're having press conferences every single day from a team of people, including the president, and of course Congress gets in on their side of it too, saying what they're going to do to help us to get antivirals and to make a vaccine. But as smart as Tony Fauci is and all these other wonderful people who are working so hard, we don't have it. We got trouble right here. I was going to say in River City, but this is America. This is the world. I don't remember how many countries, but certainly over 100 by now. So we have the SARS-CoV defined. It's enveloped, positive sense RNA virus with a genome of 29.7 kilobytes. I think that's what KB there would mean. So it's enveloped. Now, enveloped viruses, I've been learning from Vincent Racaniello from Colombia. He's a wonderful guy. Has a course online, and he's been pretty current in his 2002 lectures that are being provided, and I'm working through them, explaining that viruses that infect humans, or animals rather, have an envelope that have some sort of a lipid layer because they need to merge with the lipid layer on the surfaces of both on the outside surfaces of our cells, and then if they happen to need to get into the nucleus to do something, they also need to merge with a nuclear membrane. And when you have lipids, or fats, or I guess, we'll just call them lipids, that's the right term, they can merge with another lipid membrane very easily. Now, as an example, mitochondria in the cell have a lipid membrane, and that will often, the mitochondria will divide, and sometimes they will, instead of dividing, they'll merge, and so you have quite a bit of activity that way. When you have a lipid membrane, you can just push it together, and it suddenly joins together. We're talking about the proteins that are in there. That we remember we had the spike and the membrane protein and the envelope protein and the nucleocapsid protein, the N protein. So we have they participate in the replication of the viral genome. That's the genetic material, and transcription of structural proteins. So the spike and the envelope, the membrane, and the nucleoprotein. Now here they're calling it nucleoprotein, but the fellow from Columbia calls it a nucleocapsid. So all coronaviruses encode a set of gene-specific proteins and so forth. So I'm not going to read the whole paper to you because, well, quite frankly, you'd go to sleep and I wouldn't understand everything anyway. So, but we do have up here, it says the SARS-CoV protein, the E protein, contains 76 amino acids and is composed of a short hydrophilic amino terminus followed by a hydrophobic region and a hydrophilic carboxy terminus. Now, the reason why they're talking about hydrophilic and hydrophobic, hydro means water, and philic means loving, and phobic means avoiding or hating. So when you have a protein that has parts that are, when you have a protein that has parts that are both loving water and hating water, it tends to cause the parts of that protein to flex or to change its shape based on the exposure to water or to lipid materials. And again, membranes are made from lipids. So if you have a structure, a protein, that has part of it wants to go where the lipids are, part of it wants to go where the water is, it's going to basically stretch through or into a membrane, like a cell membrane or the nuclear membrane. And another thing about this is it forms an alpha helix. I'll just go back here. It talks about the hydrophobic region forms at least one amphipathetic alpha helix that oligomerizes to form an ion-conductive pore in membranes. So just to go back here for a second, when you have, imagine at Christmas time you got a ribbon on a package, and you take a scissor and you curl the ribbon, so you have ribbon that curls, and it kind of makes us I'll call it a curled ribbon shape. Now, I'm just going to say corkscrew, but it's really more of a curled ribbon shape. And you find those in proteins. And when you have that kind of curled ribbon shape, it goes around and around and around and around. But up through the middle of that is an ion channel. Now, if that particular part of the protein pokes itself into the lipid because it doesn't like water, in which it doesn't like the fluid that's outside in the cytoplasm or inside the nucleus, then what you end up with is this nice little, I don't call it a drinking straw, but it's like an ionic drinking straw. I don't know if it's scientific or not, but that's how my pea brain understands it. So just to say there's a lot to this deal. But the reason we're talking about the E protein is because of what they found out in the research. And let's see if we can dig through a little bit more of it, see how we do. So the requirement for E protein in virus production varies among coronaviruses. So some of them need this more than others. The SARS-CoV apparently doesn't use it the same way that some of the others do, like the MERS. That's the Middle East Respiratory Syndrome. It appeared, I think, around 2012, if I remember. So it's another coronavirus. But it was one of these deals went from bats to camels to people. I mean, that's what they're thinking. And it was called a Middle East Respiratory Syndrome. But of course, today it's unpopular to say anything about where this virus came from. So we'll just have to say the virus came from nowhere. And of course, we call it, SARS-CoV-2. That's the politics of today. Let's see. I don't know if you like that airplane above here, but that's what we got. We're in the flight path sometimes, so we're going to have some of those things going on. So we got, let's see. Anyway, so the BALB-C, which is the mice they're using in this research, are infected with a mouse-adapted SARS-CoV. So what they do is, again, they're trying to, let's see if I can find, it says they started to show that the SARS-CoV-E protein reduced apoptosis and the stress response induced after SARS-CoV infections. So because they had two versions of the virus, this is again a mouse adapted version of the virus so that it would work in mice and it would cause the same kind of things in the mice that it's causing in people. The one that had the regular genome and the one that had no part of the genome that would actually code to make the E protein, got a different result in the mice. That's the point they're trying to make. And we'll go over that as we go through. Back to this. So we have this model. I guess you can see that. This model reproduces many aspects of the human disease, such as rapid and high viral replication in lungs, viremia, which I believe is the virulence of the infection, or something like that, dissemination of virus to extrapulmonary sites, accompanied by lymphopenia. So lymphopenia would be a reduction in white blood cells, and neutrophilia, which would be, I believe, something to do with increasing affection. I mean, that's what the Greek would mean for neutrophils. But I'm not sure. Maybe I got that wrong. So, and pathological changes in the lungs and lethality. So we're saying that this virus in the regular form is quite dangerous and does a lot of bad things to people, and people get these terrible lower lung infections, which we call pneumonia, and then they get filled up with fluid and they can't breathe, and then they have to be put on a ventilator. And maybe they have a ventilator available, maybe they don't. So it becomes a real challenge for trying to keep people alive. And then further, I'll go back to the other part of this. Furthermore, the disease is more severe in aged mice as occurs in humans. So that's what we're talking about here. We're saying that old people, like me, or older than me, like my mom, if they got this disease, they might have a really tough time with it, and we don't want that. So that's why we're talking today. We're discussing what happens to mice, and maybe what happens to men and women, of course, and mankind. I guess we have to call it humankind these days. But yeah, I think you understand. So I'll just go back to reading here. So activation of NF-kappa-B is a hallmark of most infections, including those caused by viruses, leading to protective and pathological responses. Now, here we have this inflammation causes a reduction in expression of type 1 interferon. Now, that would be something that would be, interferon would act to both fight the virus inside the cell, and also would signal, as it exited the cell, it would signal for the immune cells, the innate immune cells, to respond, to come back to attack the site of the infection. So we have this mouse adapted with the SARS-CoV lacking the E protein, induced less lung pathology, mediated with decreased expression of inflammation mediators, so like IL-6 as an example, lower numbers of neutrophils in lung infiltration. So I guess I'm not sure what that means, but anyway, and limited NF-kappa-B activation. Oh, okay, so this is again the one without the E protein. You get fewer numbers of neutrophils putting themselves into the lung, and less NF-kappa-B activation. That's easy for me to say. So drugs that specifically inhibit NF-kappa-B signaling led to a reduction in inflammation and lung pathology, and increased mouse survival after infection with SARS-CoV. You get it? Inhibition of NF-kappa-B helped the mice survive. These results indicated that NF-kappa-B activation is partly pathogenic in SARS and perhaps COVID-19. We're reasoning, it's as close as you're going to get to a virus that's been studied already. It's about 80% of the same genome, and it's the very closest one that resembles the SARS-CoV-2. So that's why I'm reading this paper. So, and that NF-kappa-B inhibitors are promising antivirals for treatment of this infection and perhaps for other infections caused by human coronaviruses such as MERS-CoV. Wow. When you think about it, stop and think. We have 785 of these that were identified some years ago when that paper was published. Remember we showed you that paper with all those stacks and stacks of inhibitors? And some of those are going to be effective, perhaps, with COVID-19, are going to make, not to cure the disease, but to make the disease less virulent, less powerful, to keep us away from having to go to intensive care, maybe even out of the hospital, and certainly it's going to probably reduce the amount of ventilators that we need to treat people that are really, really sick. I remember hearing a story about how they were using this with a severe acute respiratory syndrome, the SARS syndrome. And for the people who were really bad, they would have the ventilator, but then they would actually have to put the person prone for maybe 19 hours a day in order to make this thing work the best they could, just to save their lives. Yeah, it's a serious thing. So, I'm just trying to find what I can and get the information out to people. Let's see, we got a little bit more to talk about here. Let's see, we go back to the paper, and I'll move this up a little. So pathology induced by the SARS-CoV, that's the mouse-adapted 15, and then delta would be without the E protein in the BALB/c mice. That's the headline on this results. We previously showed that the mouse-adapted SARS-CoV, the R SARS-CoV MA15 delta E, is attenuated in young 6-week-old and aged 12 to 18 months old BALB/c mice, leading to no significant weight loss and no mortality. So that's what they had shown previously in their discussion. And then the regular SARS-CoV, mouse-adapted version, caused intense lung inflammation, characterized by a number of things. So it's bad news at Black Rock for these mice if they had the regular SARS. Anyway, I think we could take a look here. So what are the points that are the main points to consider from this paper? Mice who are infected with a similar coronavirus to SARS-CoV-1 get sick, really sick. They die. If they get the version without the E protein, they do much better. Also, inhibiting NF-kappa-B seems to be a good thing under viral attack. But we do not know which inhibitors help properly. We may be able to figure out which are good if we look at other papers which reveal natural product formulas used to help protect hospital staffs who served under the first SARS attack in 2002-2003. And there are papers that discuss which natural products were taken by those people, and they compare that group of people against a larger group of people who didn't take those things. And we'll discuss that another day, but I'm just saying there is some real information there that we need to look at. So getting to know more about the E protein can guide us to find ways to inhibit its activity. And here are some conclusions from the paper. So I'm just going to do some more reading, but it'll be on this screen instead of on the paper, the white paper. So interestingly, the authors say, pulmonary pathology was clearly lower in infected mice treated with each of the NF-kappa-B inhibitors. Now, they actually used 2 initially, and then they used 2 additional ones. Furthermore, the higher reduction in pathology was observed in the mice treated with both inhibitors at the same time. So they had a CAPE inhibitor, which is caffeic acid phenylethyl ester, and parthenolide. And parthenolide, I believe, exists in an herb called feverfew in about 0.7%. And these results indicated that NF-kappa-B inhibitors also reduced the expression of pro-inflammatory cytokines and lung pathology in vivo, that means in the mice, suggesting that they may be useful for SARS therapy. Furthermore, SARS-CoV-E protein is a viroporin. Remember we talked about how it has that little curled ribbon shape and that it loves membranes that are made of lipids. So it makes a pore. So it's a viroporin with ion channel activity. So it has a charged ability to move ions through that channel from one place to another. What would that trigger? It might trigger some activities like taking apart things or putting together things. Viruses, because they don't have a brain, they have these sort of automated things that happen when they change various charge states or pH and so forth. So we showed, rather, as the writers continue, we showed that SARS-CoV, in which this activity was abrogated, lost its virulence without reductions in virus titer. Now, virus titer has to do with how many viruses were found. Were there, I've forgotten what, let's say 10 to the third or 10 to the 10th or whatever it is. So the virulence was reduced, but with the same number of viruses. Again, indicated that an intrinsic property of the E protein is responsible for virus virulence, and that changes in virus titer were not necessary for increased attenuation. So you want less inflammation, you want less sickness, you want less death. You don't have to necessarily knock off the number of viruses. You just have to knock off the effect of the E protein. That's my 50-cent version of this. Inflammasome activation was observed in cells infected by these viruses when the viroporin displayed ion channel activity, but not in its absence. Also, expression of E protein from porcine reproductive and respiratory syndrome virus, PRRSV, in the absence of virus infection, altered ion channel concentrations within the cell, leading to inflammasome activation, indicating that the viroporin itself was responsible for the activation. Wow. Inflammasomes, they don't sound too good, but I kind of think what they are is something that makes a body, that makes stuff inflamed. Inflammasome. That would be a terrible name for a heavy metal band, wouldn't it? Inflammasome. Ah, we won't go there. But more importantly, when mice were infected with influenza viruses in which the ion channel activity of PB1F2 protein was intact or abrogated, virus titers were similar. In other words, the number of viruses were the same, and a virulent or attenuating phenotype was observed. So when it's there, virulence, and when it's abrogated, and it's toned down, attenuated. Good thing. Overall, the data indicate that the intrinsic activities of these viroporins may also be responsible for virus virulence. The authors go on to say that after infection of mice with the SARS-CoV-MA, that's mouse-adapted 15, increases in the expression of the inflammatory cytokine TNF, of chemokines CCL2, CCL5, CXCL1, CXCL2, and CXCL10, and of interleukin 6, and in the migration of neutrophils to infected lungs were observed compared to levels in the SARS-CoV mouse-adapted delta E, in other words, without the E protein, infected mice. In the lungs of human patients with fatal SARS, elevated levels of IL-6 and of chemokines such as CCL2 and CXCL10 were also detected. Boy, that's a serious sentence. That's a very important message we got to think about. We got to work on this. So I'm inviting the world to just get on board here and figure out something. 785 of these guys. Some are going to work better than others. We got to work on it. And they go on. NF-kappa-B was differentially activated following SARS-CoV-MA15 delta E and SARS-CoV-MA15 infection in cell cultures and in vivo. This transcription factor regulated the expression of pro-inflammatory mediators such as TNF, CCL2, and CXCL2 after SARS-CoV infection. Administration of two inhibitors of NF-kappa-B, that's CAPE and parthenolide, increased the mouse survival after infection, and was associated with reduced expression of pro-inflammatory cytokines in the lungs. These drugs showed complementary effects as treatment, with both of them increased survival to a higher extent than treatment with either one alone. Think about it. Those things exist. Maybe we should be trying them. Hey, a lot of words, a lot of things to think about. So how do CAPE, that's caffeic acid phenylethyl ester, and bartholonide work? And which other inhibitors work similarly? Well, we need to look into that. Where does the E protein work in the cell? Where does it do its activity? Remember, we have to grab some envelopes from someplace because viruses do not make their own lipid membranes, but they do, especially if they're working with animals, infecting animals, they're going to need them, so they borrow them from someplace. So we need to discover where that E protein is doing its job, and people know, and we'll just cover it at another time. Can we see manufacture of interferon in the cell go up and still maintain the quiet neighborhood approach of reducing IL-6, TNF, CCL2, and CXCL2? That is an interesting thing, and I think there's an answer. And what natural products that exist in herbs, as an example, feverfew, that's easy for me to say, feverfew that contains parthenolide, that people have already been using for centuries that might be useful here. Maybe we can find things that exist already that people can take, and we know they don't make them sick to take them, that they're going to help them. What a concept. So, the best laid schemes of mice and men go off to glee. But we need to come back to thinking about how they might not go off to glee. They might actually work. And I'm just sitting in my studio talking, reading papers. I bet you there's somebody with the right tools that could take a look at this, and maybe a natural products person, maybe a Chinese scientist. Who knows? But I'm recording this March 19th, and I just hope that we get a handle on this pretty darn quick. We need to help people. And we need to help our whole world. The economy of the world really is tanking right now. And we're doing it for the right reasons. We're doing it to keep from hurting others by getting infected, keeping away from infection. And our world has changed, and it will be affected for a long time, perhaps decades. If you think back to some of the other things that happened in world history, I think of, in my family, the Great Depression. How in 1940, one of my grandfathers couldn't find work for a long time and finally was put to work by the WPA, the Works Project Administration, as a bookbinder. That man I knew, and so somebody that I treasure. I'm wishing all peace, and may you live a healthy 100 years. Thank you.